Okay, so thank you everyone for coming to our journal club. I'm very pleased to introduce Carol Netkoven. Carol is a researcher at the University of Cambridge, and she's working on how different regions of the brain work together to lead to complex behavior. And she got her PhD at Oxford, which is really cool. So today she'll be talking to us about the role of GABA in human motor adaptation. Without further ado, I'll let Carol begin her talk. If you have any question, please raise your hand or type your question in the chat and I'll answer them. Try to ask Carol to answer them, hopefully at the end of the talk. Yeah, thank you everyone for coming. And Carol, please begin.
Great, thanks so much. So thanks for the introduction and for this opportunity to present my work. So yeah, I'm right now a postdoc in the Bulmer group working with Sarah Morgan in Cambridge, but I did get my PhD at the other place, so I'll be talking about that work briefly today. And specifically, I did my PhD on the role of GABA in human motor adaptation, and I'll present one of the projects that links GABA, corticocerebellar connectivity, and features of motor adaptation. And I'd like to begin by just telling you briefly why I'm interested in motor adaptation and why we should get a better understanding of how the human motor system adapts. So when we move through our world, we're constantly confronted with changes in our environment and our bodies that we need to adapt our movements to. So for example, our muscle properties change as a result of development or exercise or disease, and also the objects that we interact with have different properties that can change. So they can change in weight, size, or how well we can manipulate them. And in order to smoothly interact with our world and keep up performance, we're therefore constantly required to adapt to these changing conditions. And this motor adaptation process goes largely unnoticed, but is actually so important that the human nervous system seems to have a dedicated mechanism for it. And although we have a good idea of which brain structures are involved in this kind of motor adaptation, we don't know yet precisely how they interact to adapt our movements and to store this adaptive movement. So in motor adaptation, we know motor adaptation is a fundamental process for the human nervous system. And as an illustrative example of this kind of process, I'd like to bring up something we use every day, a computer mouse. So since you've all had to work from home lately, you might have had to get used to using a different computer mouse. And so in the beginning, you'll maybe have to get used to these changed properties of your home mouse and make errors, which then you gradually decrease as you get used to these new properties of your mouse. And this means you have adapted your movements to those new properties, so something we do almost every day. And then once you might return to your department mouse, you will again make movement errors, this time in the opposite direction. And that is because you've retained some of that compensatory movement, so you've stored some of that adaptive movement. And so just to get terminology out of the way, in my talk, I'll be referring to the phase where people change their movements in response to a perturbation as the acquisition of an adapted state. And I'll be referring to this phase where the perturbation is removed after some time and people display an after effect that is gradually reduced as the retention of an adapted state. So one framework of adaptation suggests that the acquisition of an adaptation task primarily involves the cerebellum, but that the retention of this adaptive movement relies on M1. So evidence for this comes, for example, from a brain stimulation study where it was shown that, we can see in the cerebellar stimulation group, when we stimulate the cerebellum, people adapt more quickly to a perturbation. And there's no effect of M1 stimulation on this acquisition phase. But then when we look at retention of this adaptive state, actually it's the M1 group that shows increased retention of this adapted state. And this kind of framework is in contrast to other forms of motor learning, such as motor sequence learning, which is thought to primarily involve M1 at the acquisition stage. And in motor sequence learning, we know that the acquisition of a motor sequence is thought to involve M1, and that GABA concentration measured from M1 correlates with the change in reaction times due to sequence learning. So GABA is important for motor learning, and these and the M1 GABA concentration at baseline correlates with how well people learn on a motor sequence learning task. So we therefore hypothesize that in adaptation, where M1 is involved in retaining the adapted state, early M1 GABA concentration would correlate with retention. And secondly, it's been shown that functional connectivity between left M1 and right cerebellum changes after performing an adaptation task. So specifically, the activity in left M1 and right cerebellum becomes less synchronized with the activity in left M1 becomes less synchronized with the activity in the right cerebellum. You can see these pre-time courses here before the adaptation task, and then the post-time courses right here after the adaptation task. So we therefore also wanted to test whether retention would correlate with this change in connectivity between left M1 and right cerebellum. So in order to test this, we conducted a study where we got 15 healthy right-handed participants to attend 2 MRI sessions, where they were scanned either during a rotation task or doing a control task. And I will show you the rotation session first. So inside the scanner, participants controlled a cursor on a screen using a joystick with their right hand. They were instructed to hit 1 of 8 targets, one of which you can see here in green. And this would be the optimal movement, and this might be the participant's suboptimal movement. And these were the baseline trials during the baseline phase. After these baseline trials, we rotated visual feedback by 10 degrees. So this is, again, the optimal movement, the participant movement, and this is the visually rotated feedback that the participant sees. And then we go on to increase that rotation by another 10 degrees every 40 trials, such that we end up with a rotation of 120 degrees by the end of the experimental phase. And then outside of the scanner, we took the rotation away to assess retention. Now, during task performance, in order to assess M1 GABA, we collected magnetic resonance spectroscopy data from a voxel positioned over the left M1 hand area. And to assess connectivity changes, we collected resting state fMRI data before and after the rotation task. Now, the control session looked exactly the same, just with the only difference that there was no rotation imposed at any point during the session. So participants just shot targets throughout the session unperturbed. So I'm showing you here the MRS blocks acquired from the left M1 hand area. And each one of those MRS blocks consists of 53 individual spectra, which we then averaged to increase SNR. And the MRS block acquired at the baseline during the 0-degree rotation trials serves as a measure of baseline GABA. And then on the right, you can see just an overlay map for the voxel placements for all the participants in the rotation and the control condition. And we saw that the MRS placement was quite consistent, so there was no significant difference in how well we covered the M1 hand area. And then below that, you can see an example spectrum that we get out, so a raw spectrum right here. And in order to obtain our measurement of GABA, we fit a linear combination of all the spectral contributions of all the different metabolites that we expect to be there in the voxel to that raw spectrum. And you can see the fit right here. And from that fitting procedure, we then obtain our GABA measurement. And we then exclude those GABA measurements that didn't meet our quality assessment criteria, but for the baseline measurement, actually all measurements survived. Then I'm just showing you here the behavior that was acquired. So you can see participants fluctuating at around 0 in the baseline trials. And then as soon as we impose the rotation, there's this offset which participants adapt to. So you can see this stepwise adaptation pattern. What you can also see is that once we take the rotation away outside of the scanner, participants show this pronounced after effect. So just to tell you how we quantify behavior, we got our adaptation measure from the angular error, so the averaged angular error in these rotation blocks. And we get our measure of retention performance from this early phase of the washout block, so by calculating the mean error during this early phase. So before actually testing our specific hypothesis on M1 GABA, we wanted to see whether our adaptation paradigm drives changes in functional connectivity in the cerebellum. And that's because this adaptation paradigm is kind of unusual in this use of a stepwise increase in rotation. So we wanted to make sure that the adaptation task actually drives changes in the cerebellum that are known to occur in response to adaptation. So specifically, we set out to replicate this paper by Albert and colleagues, which shows that after adaptation, but not after control, you've got increased functional connectivity of the cerebellar network. So to test this, we used a similar approach. We used, as Albert and colleagues did, we used ICA to derive resting state networks. And we then tested whether cerebellar connectivity differed between the post-rotation and the post-control scan. And in our experiment, that's indeed what we find. So first of all, you can see in magenta, that's a mask of the actual full cerebellar network. And then overlaid on top of that, you can see in these red-yellow voxels, those are the areas where there's increased connectivity after adaptation compared to after control. And then on the right, I'm just showing you the same results, these same red to yellow voxels, just overlaid on the cerebellar flat map, so you can see this a bit better, just spatially where this occurs in the cerebellum. And what we can see is that the areas of increased connectivity match quite nicely the somatotopic representation of the right hand in the cerebellum. And then we actually also tested the specificity of this result, just seeing whether there's an increase in connectivity in a control network where we don't expect an increase in connectivity to happen, the default mode network, and there was no increase of connectivity in that network. We also extended the analysis from the Albert paper to see whether, if we look inside the rotation condition, this change in strength of the cerebellar network from pre to post correlates with the actual performance on the adaptation task. And that's what we see. So we see that people who adapt very badly show more of a decrease in the strength of the cerebellar network compared to people who perform quite well on the adaptation task. So you can see the errors on the adaptation task are around 0. Those show more of an increase in the strength of that cerebellar network. And there's no similar relationship for the change in strength of the default mode network. So that's specific to the cerebellar network. We also modeled the adaptation performance using a single state space model. That's a very common approach used in to quantify adaptation. And we just wanted to make sure that our results don't depend on the way we quantify adaptation. So this is just to show that, first of all, the adaptation error measure, so the model-free measure of adaptation and the adaptation parameter that we get out of the single state space model, they correlate highly, so they seem to quantify the same thing. And also when we substitute adaptation error with adaptation parameter as a measure of adaptation, then our results regarding the change in strength of the cerebellar network hold. So again, the better people adapt, so the higher their adaptation parameter in this case, the more likely they're to show this kind of increase in the strength change of the cerebellar network. Right, so now we actually went on to test our specific hypothesis regarding early M1 GABA concentration and retention of the adaptive movement. So we used Spearman's correlation to test for a correlation between baseline M1 GABA concentration and retention. And we find that M1 GABA concentration at baseline correlates with retention and not with adaptation. So higher concentration of GABA at baseline is associated with greater retention of the adaptive movement. So greater retention in this case is indicated by a more negative error because people are coming from down below there. They have the more negative their error is in the washout phase, the more they've retained of that adaptive movement. And you can see there's no relationship between M1 GABA and adaptation. Next, we tested whether the change in M1 cerebellar connectivity correlates with retention. And that's also what we see. So we see that people who show more of a decrease in the connectivity between left M1 and right cerebellum, those are also the people that retain more of the compensatory movement. And again, this is specific to retention. There's no relationship with adaptation here. And then finally, we tested whether there's a relationship between M1 GABA and M1 cerebellar connectivity change, and see that people that have higher M1 GABA concentration at baseline also show more of a decrease in M1 cerebellar connectivity. And this is specific to GABA, so there's no relationship with glutamate, for example. And although we use Spearman's correlations for these analyses, we just wanted to make sure that our results are robust against outliers. So we therefore checked all of these results, whether all of these results hold with robust correlation methods that are suitable for univariate outliers. So we used percentage bend correlations, and that analysis confirmed this pattern of results. So because we got this threefold relationship between M1 GABA, connectivity change, and retention, we then went on to explore whether connectivity change might mediate the relationship between M1 GABA and retention. So just briefly, a mediation analysis tests whether the observed direct relationship between two variables, so M1 GABA and retention, might be mediated through an indirect pathway, in this case, M1 cerebellar connectivity change. And that's what our mediation analysis shows. So it shows that the relationship between M1 GABA and retention is fully mediated by M1 cerebellar connectivity change. So regarding the question that we set out to answer with this experiment of what is the role of M1 GABA in retention, this is the model that emerges. So higher M1 GABA concentration before adapting relates to a decrease in M1 cerebellar connectivity, which in turn relates to more subsequent retention. So just to summarize what I just showed you, we find a relationship between early M1 GABA and retention. We find that this relationship is mediated by M1 cerebellar connectivity change. And I also showed you that our task drives adaptation-related changes in cerebellar connectivity. So with that, I'd like to thank everyone for listening, and my lab and my supervisors for their support. And I'm ready for your questions now.
Yeah, thank you so much, Kara, for your very, very amazing and interesting talk. So while other people are typing their questions, I will just start off and take this opportunity to ask a quick one for myself. So I'm Izu, that's my name, and I work in toxicology. So my field is more neurotoxicology. So I was wondering, my question is basically the many drugs are affecting the GABA receptor as well. So I know that you haven't showed this kind of data, but do you have any hypotheses on how, for example, drugs affecting the GABA receptor might affect behavior?
That's a really interesting question. So actually, my lab group in which I did this work, they're actually testing right now the effects of baclofen on this type of behavior. So they showed actually last year, and I think it might be already published now, that baclofen impairs, giving baclofen to participants, healthy control participants, impairs the retention on this task. And now they're looking at broader effects of baclofen. So yeah, there's definitely that idea that these pharmacological manipulations of GABA have an effect on the motor learning process, which obviously then asks the question of, well, can we manipulate? How well can we prone people to learning more effectively, more quickly, and basically employ those pharmacological manipulations?
Yeah, thank you. So I'll stop with my personal questions, and so I'll read off a question from Edit, and if you want to ask a question yourself, feel free to uh, let me know in the chat as well if you want to speak yourself. So the question is, can you use the scientific output for any disease biomarker?
Disease biomarker. So are you referring to whether this helps us? I'm not quite sure. Yeah, could you maybe specify that question further, exactly what you mean?
So I think, well, edit types, maybe in terms of connectivity, is there any psychiatric diseases uh, like, or dementia? She mentions, he or she mentions
Well, so
Alzheimer's. If
first
there's
of all,
some.
Yeah, so I think I wouldn't interpret this this result that we're showing here as a some kind of trait that we've looked at, because essentially what we're saying with this study, or what these results are showing us, are that M1 GABA, when we measure it on the day, that determines, or that has a relationship, doesn't determine, but that has a relationship with the performance, the retention performance on the day. But I've actually looked at, because this was a within subject study, I actually looked at whether this is a state or trait variable that we're looking at right here, but there's no relationship between the ammonia GABA concentration measured on the other day to the motor performance on the day of the rotation paradigm. So that tells me that this is not something that I would call a trait variable, but actually a state variable. So I wouldn't postulate that this is something we can use as some kind of biomarker for how well people are generally at performing motor tasks. So I wouldn't go that far.
Cool. Thank you so much for your answer. I'll read off the next question by Mary. So she asks, what is the repeatability of the GABA measurements in the control arm, and how does that relate to the size of changes you are seeing?
So the GABA measurements, in general, there is a quite a good reliability of GABA measured with these MRS techniques. So I think that's where you're getting at. What's the repeatability of the GABA? I think that's what you're asking. And how does that relate to the size of the changes? Do you mean the size of the changes of the connectivity?
Sorry, no. Can I? Do you hear me? Is that?
Yeah.
Yeah. I mean, you're quoting GABA to creatine ratios and how they change. You know, you're showing what the change is, but not what the actual absolute figures are at any time. And I mean, aren't you doing a sort of serial? You're doing serial measurements of the GABA over creatine.
Yeah.
But what's the sort of fluctuation from time point to time point?
Okay. So we actually, I think I have this figure right here. We actually did not see a fluctuation that differed between the rotation and the control condition. So I think I've got this here. So you can see there is quite some variability, but there's no significantly different time course that GABA takes during these conditions. So what my results were referring to was the GABA concentration at baseline, just measured at baseline on the day where people did the rotation condition, and the change was actually in the connectivity between left M1 and right cerebellum. So there was a relationship of M1 GABA at baseline, then with the change of connectivity and the behavior. But there were there was no different time course that GABA took in the rotation compared to the control group session. Sorry.
Cool. Yeah, thank you, Mary, for your question. I'll go to the next one, if that's okay. So Nashia asks, is there any advantage of doing the task inside the scanner as opposed to outside if we're looking at correlation between GABA and task activity? So does MRIs capture real-time changes in GABA?
Yeah, that's a really good question. And I think that's just getting also at this plot that I'm showing right here. So yeah, so one thing that we were interested in was whether there were changes in GABA during the course of the task, which for this task that really employs the cerebellum at the acquisition phase, you wouldn't necessarily expect. So I can kind of understand why we didn't get these changes, but that was something that we wanted to look at. Because that has been shown. I think I've also got a slide for that. That's been shown with motor sequence learning. Where do I have this? Oh, I don't have this. No, I think I've got it right here. So for motor sequence learning, it's been shown that actually when you learn a motor sequence, you've got this reduction of GABA compared to when you just do a control task or you just rest inside the scanner. So you can actually see these real-time changes, as you call them, over, I mean, it is a prolonged period of time just because you need the SNR to be able to reliably quantify GABA, but we can see these learning-related changes in GABA in M1.
Cool. Thank you very much. So there's another question. I think we'll make it one of the last one because it's a nice concluding question. So Edith asks, what is the next step? What's the future?
Yeah, so the next one I actually already took during my PhD. As you might have guessed, I was really interested in also looking at cerebellar GABA. And so we did that. So we did cerebellar MRSI, which is difficult to do, but we achieved good MRSI resolution in the cerebellum, looking at the time courses of changes there. And then the ultimate goal, what's also really interesting, is looking at interventions that once we know, so what's the kind of natural time course of learning that GABA takes during kind of natural learning. And then the second question is, well, can we drive this? Can we employ interventions, pharmacological interventions, but also specific interventions that target a specific region, like CDCS, for example? Can we use this to drive these GABA changes and then improve performance? So this is all kind of looking also at motor rehabilitation applications. So yeah.
Thank you so much. So on this point, and in the interest of time as well, I will just like to thank Carol again and everyone for attending, and the people who asked their questions. Thank you very much. So thanks everyone, and we hope to see you at our next journal club. So I'm Izu, and thank you and have a nice day.
Bye.